Hello. Tonight we're going to talk about structure and function of cell membranes. One thing AP does that's really important is that they often ask questions about structures and functions and how those affect the organism. So specifically we're going to talk about membranes tonight. The model that they use to describe a membrane is called the fluid mosaic model. What this means is it's an understanding of cell membranes that the membranes are fluid, which not fluid like liquid, but it means that they're not stationary. They move. There's proteins embedded in the membranes, and they move around and change position all the time. So here's what a membrane looks like. You've got all kinds of proteins, cholesterol molecules. You've got some recognition molecules coming off. Let's stop for a moment and watch a little YouTube.
Of the inner cell and the harsh conditions of the outside world stands the cell's plasma membrane. As crucial as this barrier is, it's surprisingly flexible. Push it and watch it move. Poke hard enough and it might break and begin to regroup. The lipid molecules of the membrane naturally assemble in a double layer because their tails repel water as their heads attract it. Throw in some cholesterol and a few carbohydrates, and you have the basic structure of a plasma membrane. Within these lipid molecules, we also find different proteins which do various things for the cell. For instance, they receive signals from the world outside, or they transport nutrients and waste. So nature composes the membrane with a combination or mosaic of different lipids, carbohydrates, and proteins. And these molecules are not stationary. They constantly move within the structure, fluidly changing their positions. The survival of all life rests on this veil of material. A supple membrane just 2 molecules thick.
Gives you an idea what cell membranes are kind of like. They mentioned in the video that cell membranes are made from a phospholipid bilayer, which means you have 2 phospholipids, one with their tails facing each other. Now the tails are hydrophobic, which means they repel water. They hate water, literally. Hydrophilic heads on the outsides love water. And so they line up, like in the video, with their heads on the outside and on the inside, and the tail regions facing each other between the layers. The phospholipid bilayer also contains cholesterol. They mentioned that. What that does is it makes the bilayer stronger and more flexible and a little bit more permeable to lipid molecules. The plasma membranes are selectively permeable, which means they allow certain materials to enter the cell and other materials can't enter the cell. So they select what they're permeable to, what they will allow in and what they won't. They're made primarily of the phospholipid bilayers, because there's lots of phospholipids, and then they also have proteins embedded in. There are 2 basic types of proteins. Integral proteins, meaning they go all the way through, so this is an integral protein, they go to both sides, and a peripheral protein, which is just on one side. This is another peripheral protein. The cell membranes have several functions. First of all, they control what goes into and out of the cell, and then they communicate with other cells, like sending hormones out or neurotransmitters. Those are forms of communication. They have proteins embedded in the outside, which can be used for recognition. White blood cells, use those to know what cells are normal to your body and which ones are bacteria or viruses and need to be killed. You also have transport proteins that regulate the movement of water and soluble molecules through the membrane. So there's lots of functions for the cell membrane, and we're going to go into several of those very specifically. I think one of the coolest things about cell membranes happens when material is coming into or out of the cell. Now, I'll come back to this slide. Let's look at the picture, because I think a picture's worth 1,000 words. If this is material that ribosomes made at the ER, they made those little molecules, they bud off into a vesicle, the vesicle comes, fuses with the Golgi, it's going to be processed, and then it forms another vesicle. That vesicle comes to the cell membrane, it fuses with the cell membrane, pushes against it and ruptures, and then that vesicle membrane becomes cell membrane. And likewise, when material comes into a cell, if I'm going to phagocytize some food or some large molecules, those molecules push against the cell membrane, creating a vesicle, which then can go and fuse with a lysosome and be consumed. It can fuse with a Golgi to be processed further. Several different things can happen. But the cool thing about the membrane is that the membrane is recycled. It comes in, it goes out. You know, the inside of a vesicle membrane is really the outside of a plasma membrane. So let's go back to that slide, and that's what I wrote. So it's really kind of cool. I mentioned several parts of the cell membrane. The phospholipid bilayer, we know about that. One type of protein is called a channel protein, and what they do is they create a tunnel for one specific protein to come through. This part A is a channel protein. There are other proteins that use ATP to move molecules through the membrane. Okay, so it's going to go through active transport. You have to have the energy to get that specific molecule to come in. You can have recognition proteins. So if this is a white blood cell and this is a normal body cell, that white blood cell is going to look for a specific recognition protein that has a carbohydrate sticking out on it, and it's looking for the specific carbohydrate. It says, oh, that carbohydrate's normal to you, that cell is you. It's not a foreign body. You also have proteins. This is an integral protein, which go all the way through the membrane and allow chemical reactions to occur on the inside of the cell. So this molecule is going to come into this enzyme, create the blue square. The blue square goes into the next enzyme, creating the green circle. And whatever the green circle was, that's something the cell needed, some substance, some chemical reaction just occurred. You can also have proteins that join two neighboring cells together, like in your heart. You want all the heart cells to be joined together to create the organ of the heart. You don't want them to be separated and pump on their own, beat on their own. Your heart wouldn't function then. So you have these intercellular joining proteins that can be part of a cell membrane. You have receptor proteins. Now, what a receptor protein would do, for example, like a hormone, if a hormone attaches to the receptor protein, it causes some type of signal transduction pathway to begin on the inside of the cell. The hormone itself does not enter the cell. It just causes a reaction that then tells the cell to do something. You have electron transport proteins that will transport electrons from one molecule to another. We already talked about cholesterol. It decreases the permeability to water and more permeable to lipid molecules and helps to maintain the shape of the cell. So that's a cell again. And you can see all the different types of material that you find in a cell membrane. The last thing, which you kind of look back at that slide and you say, ooh, what's all this stuff on the outside? That's all extracellular matrix, which is a whole bunch of microfilaments and other parts of cytoskeleton that attach one cell and can attach to other cells. They help to fix a cell in a specific location, and coordinate extracellular changes and several different things. We're going to talk about how material enters and exits a cell now. Some of the terminology you need to be familiar with would be concentration gradient. That's the difference between the concentration on either side of a membrane. Usually one's higher than the other. Isotonic is when the concentration on the different sides of the membrane are equal. Is in math means equal, and so isotonic will be equal concentrations. Hypertonic means that you have more solid molecules on one side of the membrane than the other. So if I'm looking at the little circles, okay, no, let's look at the big circles. The big circles are much more concentrated on the outside of the cell than the inside of the cell. So the outside solution is hypertonic. Hypotonic is when you have less solute on one side than the other side. So in this case, there's a whole bunch of those circles on the inside. There's less on the outside. So this is hypotonic solution on the outside. Okay? Now, these two words you'll use in conjunction together, which means at the same time. The inside of the cell is hypertonic. The outside of the cell is hypotonic. There is less solute out here than in here. Okay. There are two main methods that materials move across a membrane. They go through either passive transport or active transport, and there are several examples of each of these. Passive transport does not require energy, and active transport does. The energy form is ATP. Passive transport will include things like diffusion, osmosis, facilitated diffusion, and gated channels. Active transport will include the sodium-potassium pump, endocytosis, and exocytosis. So we're going to talk about each of these. Active transport, like I just said, is movement against a concentration gradient. It goes from a low concentration to a high concentration, which is backwards. So it's really hard to move a material backwards. It's kind of like rolling a big, huge rock up a hill. It's a lot easier to roll the rock down the hill than it is to roll the rock up the hill, and it takes a lot more energy to roll it up the hill. And that's what active transport's doing. It's moving the material into an area where there's a lot more of the exact same material. Passive transport moves the material from a high concentration to low, and no energy is required. You know, the rock at the top of the hill, it rolls down the hill very, very easily. Osmosis is the movement of one specific molecule, water, across the membrane from high to low. Okay, this membrane is impermeable to the blue molecules, so those molecules cannot cross the membrane. To reach an equilibrium or an isotonic solution, the only thing that can move them is the water molecules. And so the little tiny red water molecules are moving across the membrane trying to equal so that the concentration of red is equal on both sides. Diffusion is the same type of thing, but it's a different solute molecule. Osmosis is only water, and this would be a different molecule. Okay, water's the background. The blue or the orange things are dye molecules. So on one side of the molecule of the membrane, you have a whole bunch of dye. Those molecules will go through the semi-permeable membrane, and it tries to reach equilibrium. And here they show an example doing it with two different molecules. The orange is going to move this way, the purple is going to move this way. That's diffusion. So if I have a hypotonic solution and a hypertonic solution, what happens is, and this membrane is impermeable to the purple molecules, the water's the only thing that can move, and so water will move trying to dilute the hypertonic solution so that it reaches equilibrium, and you will literally get this type of thing happening in a tube. It's kind of cool. Okay, in real life, let's look at what really happens to cells when they're in a situation where it's a hypotonic solution. Okay, when there's less concentration of solutes inside the cell, and the solute cannot enter of its own concentration, then the only thing that will happen is the water will leave the cell. And thus the cell will become more and more concentrated. And the cell will either shrivel or go through a process of plasmolysis. Now that's kind of a weird picture of this one. This is solution where it's hypotonic on the outside, and I have a description of hypotonic on the inside. Anyway, ignore the picture for now, but when a cell is in a situation where there's less solutes and the solids can't leave, then the water's going to leave, and this causes the cell to shrivel up. Okay? Isotonic water is going to enter and leave at the exact same rate. A hypertonic solution is when you have a higher concentration of solute on the inside than on the outside, and so the water will enter the cell to try and dilute the concentration. When it tries to dilute the concentration, the cell will swell and it'll cause it to lyse. Lyse means rupture, and so this picture is showing it rupturing. Or in a plant cell, it becomes turgid. So let's kind of look at these two, since I didn't show you a picture of one shriveling. When normal red blood cells are put in distilled water, there are no solutes in distilled water, and the solutes, the hemoglobin inside the red blood cells, can't leave. The blood cells will fill up with water and literally lyse. They will burst. If they're put in a concentrated salt solution, so there's a lot more salt on the outside than on the inside, the salt can't enter to reach isotonic, so the water on the inside has to leave the cell. It causes the cell to shrivel up and die. Plant cell, if it's placed in distilled water, the water will come into the cell. Because the plant cells have a rigid cell wall, it prevents the cell from lysing, so the cell just becomes turgid. However, if you put it in a concentrated salt solution, the water leaves the cell and it goes through plasmolysis, which basically means the cell plasma membrane will pull back from the cell wall and the membrane inside will shrivel up, but you'll still have the plant cell wall on the outside kind of staying there. So it's kind of like the cell inside shrivels up, but because it still has the plant cell wall there, it's called plasmolysis. Just a little different term. In the previous lecture, we talked about a special organelle that paramecium have, a special vacuole called a contractile vacuole, which helps prevent paramecium, which are freshwater protists, from dying in the water, basically. They have a whole bunch of different kinds of solute molecules on the inside of their body. But in the fresh water, there's not very many solutes. So what normally would happen, and what does happen, is the fresh water will move into the paramecium to dilute it. Well, if the water moves in, the paramecium is going to lyse. It's going to die. So it collects the water in this little contractile vacuole, which has a pore on it, and then it spits the water out, spits it out, and you're left with just the little empty contractile vacuole right there. And that prevents the cell from actually lysing. Another type of protein you have is called a carrier protein, and these will carry the molecules from the outside to the inside or inside to the outside, whichever way it's supposed to go. These molecules are very specific to one type of protein. So only the little circles can enter through this tunnel. A triangle couldn't enter. It's not the right shape. Now, sometimes it causes a shape change in the molecule where it fits in a little active site in the protein, which causes a three-dimensional conformation change, making it open up on the other side, which causes it to release the molecule. Well, once it's released, that molecule goes back to its normal position. Okay, that's caused from hydrogen bonding. It doesn't need energy. Some of these molecules do the same thing, but they require ATP to do that, which would be active transport. This is passive transport. We're going to watch a short video. This video does not have narration, but it's going to show you this process. Okay, it's hypertonic out here. There's more solutes out here. So we need to bring these molecules in. They need a carrier protein to facilitate their entrance. It comes, hits an active site, which causes a conformational change. Conformational change. Which allows it to then enter the inside of the cell. Another molecule hits the active site, conformational change, and that one enters the cell. Now I have 3 circles on the inside, 3 circles on the outside. What do you think that means? It's reached isotonic situation. When it's isotonic, the molecule will move into and out of the cell at the same rate. Okay, the next one is called a gated channel. This is very similar, but it needs a key to open the door. And usually the key is a different type of molecule. Okay, gated channels can help cells to get sodium or potassium or calcium or chlorine, specific molecules that they need, but they need another molecule to help open.
When the cell membrane is at its resting membrane potential, the activation gates of the voltage-gated sodium ion channels are closed and the inactivation gates are open. Voltage-gated potassium ion channels are closed. Depolarization is initiated by a stimulus which makes the membrane potential more positive, causing the voltage-gated sodium ion channels to start to open. As threshold is reached, many sodium channels open. Sodium ions diffuse across the membrane, causing depolarization. Voltage-gated potassium ion channels also begin to open, but more slowly. Therefore, depolarization occurs because more sodium ions diffuse into the cell than potassium ions diffuse out of it. As the membrane potential approaches maximum depolarization, the inactivation gates of the voltage-gated sodium ion channels begin to close, and the diffusion of sodium ions decreases. The potassium ion channels remain open, and potassium ions continue to diffuse out of the cell. The increased potassium ion permeability lasts slightly longer than the time required to bring the membrane potential back to its resting level. The extra efflux of potassium ions causes the membrane potential to become slightly more negative than the resting value. After the voltage-gated potassium ion channels close, the active transport of sodium and potassium ions reestablishes the resting membrane potential.
Of a gated channel, that was a voltage-gated channel. There are other types of gated channels, but that's one type. That particular type is found in nerve cells, and it's how the electrical signal moves down a neuron. We'll find out more about that when we get to the nervous system. Okay, just a moment to review. Diffusion is the movement from a high concentration to a low. Facilitated diffusion just has to have an extra protein to move those molecules from high to low. Active transport is going to take some energy to do that, and it's going to go from low to high. So let's jump into active transport. Active transport takes molecules from a low concentration to a high concentration with the use of energy. A good example is the sodium-potassium pump, which is going to move sodium and potassium. Now, this also is found in nerve cells. After the voltage-gated channels move the ions to the opposite sides of the cell that we just saw in that video, then we have to move the ions back to where they started so the nerve cell can be activated again. So this returns everything back to normal. So let's take a little chance to watch this video, and then I'll describe this. Okay, on the outside, you have a whole bunch of sodium. On the inside, you have a whole bunch of potassium. So some of the sodium on the inside is going to bind to the molecule. We need to return the sodium back to the outside. So with the adjunct of ATP, it's going to phosphorylate your molecule, which causes a conformational change. That then disassociates the sodium. Sodium will float away. There's more potassium on the outside. So some potassium is going to come, hit this molecule. That causes a 3D conformational shape change, disassociates the phosphate molecule, and then the potassium will then be released to the inside of the cell. So the voltage channels took the potassium, the Ks, out of the cell. The sodium-potassium pump brings it back in. Likewise, the gated channels allowed the sodium to come into the cell. This allows them to go out of the cell. Okay, so you can stop the PowerPoint here if you would like, and this will take you through the steps of that 6-step process to get the sodium back out of the cell and the potassium back to the inside of the nerve cell. Another thing that can happen, which is found in mitochondria and chloroplasts, is called an ion pump. And what these do is, with the ATP help, it pumps hydrogen from a low concentration to a high concentration across the membrane in the mitochondria and the chloroplast into the stroma or the fluid matrix. It's across the cristae membrane to get to the inside. The reason it does this is another protein later on called ATP synthase is going to allow the hydrogen ions to come back through, and in the process it's going to create energy. But the first thing we have to do is pump the hydrogen across the membrane. So here we are pumping it across the membrane with ATP's help. You can also have a cotransport molecule, protein, that's this one right here, and this is an example of how sucrose can enter a cell. So the hydrogen ion is going to be pumped across, that's the ion pump. But then the hydrogen ion attaches to this cotransporter with the sucrose, and that brings the sucrose into the cell. That's how sucrose gets in your cell. Earlier I told you about exocytosis and endocytosis. Exocytosis is when you have a vesicle filled with something the cell needs to export. It pushes against the cell membrane, it fuses, ruptures, and all those molecules are released out. Endocytosis is the same thing, but we're going to bring stuff into the cell. So material is going to push against the cell, creating the vacuole, and there we have it. Now that, depending on what types of endocytosis it is, there are like 3 types. If it's a large particle of food, it's called phagocytosis. This occurs in white blood cells when they're eating bacteria and things, or protists eating food go through phagocytosis. Pinocytosis is when you're bringing small stuff in, very tiny, creating small vesicles. Phagocytosis, like I said, big food particles. The last one is called a receptor-mediated endocytosis. This collects a specific molecule. For example, in a nerve cell, when it releases its neurotransmitters and activates the next neuron, those neurotransmitters are brought to the side of the neuron. They are attached to these little receptor molecules, which then engulf those neurotransmitters, and the neurotransmitters are brought back into the cell in a vesicle. It recycles the neurotransmitters so that the cell can reuse them over and over again. It doesn't have to create new ones to activate the next neuron. That's kind of an important thing because it would be a lot of energy usage to create neurotransmitters continuously. So that's a receptor-mediated endocytosis. The last thing we're going to talk about is how cells communicate with other cells next door. And there are three types of junctions, or three types of connections that they can have. Four types, actually. The first one are called desmosomes. And what they do is they act like glue, and they glue cells together. Another one is tight junctions, and they also fuse cells together. Gap junctions are some channels that you find between adjacent cells in animals. And plasmodesmata are canals or channels that you find between adjacent plant cells. So those two are very specific. Desmosomes and tight junctions are found in different types of cells. So if you look at them, a tight junction looks kind of like this, and you would find it right in here. And these are desmosomes, which have a much longer type of connection, anchoring type of junction. In an animal cell, you could have little channels between. That would be the gap junctions. And if it were a plant cell, you would have these would actually be a little bit bigger, and they would be called a plasmodesmata. Okay, so that's a plasmodesmata, just bigger, found only in plants. That's it for tonight. Have a wonderful night. See ya.